My name is Robert Klein. I'm chairman of the governing board, and I'd like to welcome you all here this morning at Stanford. We have the distinct pleasure of listening to Dr. Michael Clarke, MD, a great pioneer in the area of cancer stem cells. Dr. Clarke is the associate director of California's renowned Stanford Institute for Stem Cell and Regenerative Medicine. He was the first researcher to find cancer stem cells in a solid tumor of breast cancer and discovered that inhibition of programmed cell death is essential to the growth of breast cancers. As a minimum background, saving most of the time for Dr. Clarke's presentation, which I've had an opportunity to preview, and I think you'll be extremely pleased at the discussion of the theory of cancer stem cells and the discussion of alternative views and counter views and what the potential answers to those counter views might be. But to frame this discussion, I will tell you that he received his MD degree from Indiana University. He then was an oncology fellow at the National Cancer Institute. In 1986, he joined the faculty of the University of Michigan as a professor of internal medicine and a professor of cell and developmental biology. It's great to realize that in this last election in 2008, criminal penalties were removed from embryonic stem cell research in the state of Michigan, some 23 years after he became a professor at that school. In the fall of 2005, after the passage of Proposition 71, Dr. Clarke came to Stanford University to direct the solid tumor cancer stem cell program and to serve as the associate director of the Institute for Stem Cell Biology and Regenerative Medicine. Today, we will be hearing his perspective and insights into cancer stem cells. And while listening to him, we can all hope that this theory will be carried forward in a number of breakthroughs that will revolutionize cancer treatment, potentially lead to a Nobel Prize for Dr. Clarke, but importantly today inform the board, the staff, and the audience of the tremendous value of this theoretical approach that continues to gain documentary evidence in very high-level journals, scientific journals in this country and abroad. Dr. Clarke.
Thank you, Bob, for the very kind introduction. And I'd like to thank Alan Trounsend and the board for asking me to come speak to you about this topic. And basically, Alan asked me to talk about cancer stem cells and alternative hypotheses and present these views to the board. So what I'd like to start with is that every tissue or organ in which cancers arise have a very similar cellular developmental structure. And the way this works is that there's a short-lived population of mature cells and that are constantly being regenerated over the lifetime of an organism. So as an example, the lining of your gut all the way through your lips, through your alimentary tract, is being regenerated every 7 days. Brand new cells from your lips all the way through to your stomach and on down your alimentary tract. Your blood cells are being replaced on average, depending on the type of blood cell, every 8 hours to about 2 to 3 months for the longest lived cells. Your skin is being regenerated in 30 days. So a month from now, if you were to look into a mirror, you could honestly say that you're a brand new person because every single cell you're looking at is brand new. And it goes on. And the way this system works It's a very contrived system, and for this board, this is probably, I'm sure most of you in this room probably already know these systems, but I'm going to go over it for those who don't. And basically, the way this works is there's a stem cell, which is the only long-lived cell population in these tissues. So, and it does so because when it divides, it can undergo a type of cell division that we call self-renewing, or through a process that we call self-renewal. And basically what this means is when this cell divides, it can make one or two progeny that have, as far as we can tell, similar replicative capacity as the parental cells. There's some debate in stem cell biology whether these cells have unlimited replication capacity or whether they have a more limited expansion. And again, I think the jury is probably out on that. But certainly it's difficult to measure the decline in replicative capacity. And it also can make a progenitor cell population, sometimes called transit amplifying cells, that undergo a series of cell divisions that with each cell division, they lose replicative capacity. So they essentially become progressively more differentiated with each cell division, and they lose N-1 capacity to replicate. So they clearly have a limitation in their ability to replicate. And finally, this process of self-renewal is hypothesized, and I say hypothesized because in most systems, the interaction, hypothesized to involve signals from niche cells which give the stem cell the necessary regulatory signals to self-renew. And the reason I say this is hypothetical, except in a couple of very limited circumstances. For example, in germ cells of fruit flies and a couple other systems, these interactions between the niche and the stem cells are very poorly understood. Okay, so that's the background system. Now, if you think about what I just described for a stem cell, these cells have a lot of properties of cancer cells. They have large replicative capacity. I didn't talk about that, but they can actually migrate. So a blood stem cell can migrate from the bone marrow into the blood, go to another bone, re-enter the niche, and then reestablish hematopoiesis in that site. So these cells, that's metastasis, what I just described to you. If you look at a breast during pregnancy in mammals, the normal breast stem cells will migrate throughout the breast, make the milk glands needed for lactation. And so again, these cells have migratory properties and the ability to invade into tissue, similar to what's described or what happens with cancer cells. And in fact, I'm not going to go into the details, a lot of these same mechanisms that normal stem cells use to migrate through tissues and or through the body are also used by cancer cells to metastasize. So these cells have a lot of the properties of cancer cells have a lot of property of normal stem cells. So the questions my lab are interested in are what are the molecular pathways regulating normal stem cell self-renewal, and how does that apply to cancer? And we also want to understand what are the molecular pathways that restrict self-renewal of normal progenitor cells. And as you can see, this has direct relevance to cancer also. Do tumor cells retain some of the cellular hierarchy of tissues from what they arise? And this is important because if one wants to understand self-renewal, one has to first isolate the cells that are capable of self-renewing so we can do the molecular biology and biochemistry to understand these processes. And we want to know if stem cell pathways are utilized by cancer cells, and if so, do they impact tumorigenicity and therapy? And I already alluded to evidence that pathways used for migration of normal stem cells are used by cancer cells to spread. I'm not going to talk about that, but I am going to talk about the process I'm most interested in, which is self-renewal. And are the mechanisms of self-renewal in normal stem cells what's the same as the self-renewal pathways used by cancer cells, and what are the differences? So really the most striking visual evidence that the cellular hierarchy of normal tissues is present in cancers is with a disease that we call immature teratoma, which is a type of testicular cancer. And testicular cancer, of course, is the tumor that Lance Armstrong had and was cured of. And what one sees in immature teratoma is that there are seas of aberrantly differentiated cells, but if one stains with markers, and in this case, this is alpha-fetoprotein, which marks relatively earlier undifferentiated cells, you see that there's a few undifferentiated cells in the sea of aberrantly differentiated cells. And I won't go into the data of this, but this slide was actually the slide that gave me the notion that maybe there was a stem cell population in solid tumors. Because when you take care of patients with testicular cancer and when they have immature teratoma, this is the one solid tumor in adults which is commonly cured using a drug called platinum. And what happens with platinum treatment in about a third of the patients is they have residual cancer cells. They end up being cured, and when one looks histologically at these tumors, the mature cells are still there, but the immature cells are gone. And this really, these two observations, which, by the way, I thought were unique at the time, but I subsequently found out were made by Virchow in 1855, so I was a little late on that observation. So, but not the platinum part, but the developmental part was noticed by Virchow in the 1850s. And so that really led to this model that maybe all the cancer cells in a tumor aren't the same, although some of the heterogeneity clearly is due to mutations, clearly due to environmental influences, which is what we called the stochastic model in a review written by Irv Weissman and I in 2001. But there was a possibility that some of the only a subpopulation of these cells had this unique ability to self-renew and thus could drive tumor growth and proliferation. And some of the cancer cells may lack this ability. So to test this, Mohammed Al-Hajj adapted the tools that had been developed mostly here at Stanford for blood, for isolation of normal blood stem cells, which is flow cytometry and self-renewal assays. In this case, we used immunodeficient mice. And Mohammed asked the question, if I take breast tumors, if I dissociate the cells, isolate different population of cells, are some populations in cells enriched for the ability to form tumors, whereas other populations of cells are depleted for this ability? Now, to make a long story short, the initial findings suggested, yes, in breast cancer, there were subpopulations, and I'm actually going to show you some of that data a little later. That paper created more controversy than I've ever seen in at least in my career in science. A lot of people embraced it immediately, and a lot of people said it's the craziest thing I've ever heard. And there were several arguments along those lines. Now, I'll point out, most cancer biologists are not stem cell biologists. They came into the field as molecular biologists and specifically looking at oncogenic mutational changes and worked with cell lines. And so immediately, everybody who read this came up with their own definition of a stem cell, regardless of whether they had been working on stem cells. And so To me, and this is shared by my colleague here at Irv Weissman, we independently believe that the simplest and probably the best definition of any stem cell, be it a normal stem cell or a cancer stem cell, if you will, is very simple. Can the cell self-renew? And can the cell differentiate into non-self-renewing progeny? So a very simple, basic definition. But a lot of other definitions became applied by both people who argued for the existence of cancer stem cells and who argued against the existence of cancer stem cells. So a lot of people argue that particular markers, for example, CD44, my favorite marker because it's one we've first discovered in breast cancer and has turned out to be a very good marker in many types of solid tumors, side population, the expression of a gene called LGR5, expression of CD33, et cetera. People argue that those are markers. Many people argue that those are markers or stem cells, or a word that I detest, stemness, for whatever that means. It's that word is defined by differently by everyone. But marker expression is dependent on the microenvironment from which the stem cells were isolated. So if you take blood stem cells, the most characterized stem cell that I'm aware of, in a normal mouse bone marrow, the blood stem cell phenotype is different than the blood stem cell phenotype of that same cell when it's circulating in the peripheral blood, when it's been mobilized and circulating in the peripheral blood to go to another place in the bone marrow. So even in normal stem cells, these marker expressions, you have to be very careful, and they are only useful in the context with which they were defined in the definitive set of experiments, which were measuring self-renewal and differentiation. And not all cells that express such markers are stem cells. So most cells that express CD44, CD133, LGR5, I'm not sure about because it's a newer marker that we don't know as much about, but almost all the markers used for isolation of normal stem cells, most cells that express those markers are not stem cells. Let me repeat, are not stem cells. So when you put them in stem cell assays, those cells won't self-renew, lack differentiation, or all of the above. So another commonly used definition of stem cells is the ability to form spheres in defined culture conditions. And again, this, in my opinion, is not a priori a definition of stem cells, because many stem cells cannot form spheres in published media that's been published for various assays, for example, in the breast. In fact, and many non-stem, and in the breast system, which I know well, normal breast stem cells do not form spheres as single cells in a media called mammosphere media. Only aggregates, they only form aggregates in such media. And if you look at some progenitor cells, they will form spheres in this same media. So the idea that sphere formation necessarily means a cell is a stem cell, I think is not correct. And quiescence, it's very clear in the blood, and I think there's now evidence in a lot of systems, the skin, and I think there's going to be evidence in the gut coming out that there are stem cells that are proliferating, and there's stem cells that are quiescent. These are different stem cells undergoing in a different state, probably as in response to different requirements from their environment. So, for example, if it's well known that if you stress the blood system and eliminate many of the mature cells, there's a quiescent set of stem cells which then go into cycle, start proliferating, re-expand the blood system, and once you go to blood homeostasis again, those cells then revert back to a quiescent state. And last one, recently, this process called epithelial mesenchymal transition, which is a process by which an epithelial cell undergoes, obtains some characteristics associated with fibroblasts that is a definition of stem cells. And I can say from our work, some stem cells can certainly undergo EMT, but not all stem cells have undergone EMT at any point in time. So there's clearly cells in the breast system that we've seen that express E-cadherin, very high levels, which is lost when a cell undergoes EMT. And in tumors, we have actually sorted cells that express high levels of E-cadherin and low levels of E-cadherin based on other markers that we isolate the stem cell population. And both populations of cells were equally tumorigenic. So it's clear the cells can undergo EMT, but not every cell has done so. Now, one other confusion is that when you use the word stem cell, to many non-stem cell biologists, this implies that tumors have arisen from the normal stem cell. But that's not necessarily the case. And in fact, Phil Fialco, who I think provided the first evidence for, the first solid evidence for a stem cell population in a tumor in humans, in a series of seminal papers in the mid-1970s, what Dr. Fialco found is that when he used a marker that was for an X-linked gene, G6PD, that when you look at blood cells, an individual, you can tell whether it arose from a single clone or if a group of cells arose from a single cell, then they will always express the same isoform of G6PD. And what Dr. Fialco found was that in some leukemias, there was a cell, an early cell, that was clonal, that was giving rise to post-mitotic cells, red blood cells, differentiated cells in the blood cells, cells that had clearly lost the ability to replicate anymore, that sometimes they arose from an early cell and sometimes they appeared to arise from a more differentiated cell and had a much more restricted ability to differentiate than the stem cell. And his conclusion was, first of all, there was an early cell that had the ability to self-renew in these tumors. And his second conclusion is that the origin of that cell varied from patient to patient. Sometimes it seemed to arise from a very early cell, sometimes from a more mature cell. And then I think in really a seminal paper from Irv Weissman and Katrina Jameson, when Katrina was in Irv's lab, they looked very carefully at chronic myeloid leukemia. And this is a complicated slide, but what they found was in the chronic phase of chronic myeloid leukemia, the stem cells, the only cells that were able to transfer the disease in immunodeficient mice, seemed to be analogous to a normal stem cell. So the leukemic stem cell appeared to arise from either a normal stem cell or a very, very undifferentiated cell. Whereas the majority of the cells in the disease were unable to transfer the leukemia and were not able to self-renew. But what they found in patients who had progressed to blast crisis, that those patients, even though their bone marrow looks like the cells are immature, the cell that's driving the leukemia, the self-renewing cell, in this case, actually appeared to rise from a committed progenitor cell that had acquired the ability to self-renew, But these cells still had some ability to differentiate because they would make blast cells, another immature group, but slightly more differentiated than the leukemic stem cell, that were unable to transplant the disease. And I think this was the first formal proof of, in humans at least, of leukemia stem cells arising from a progenitor cell population. Now, this is very confusing to non-stem cell biologists because you've got a progenitor cell that's acquired the ability to self-renew. And if you go back to my initial definition, it fits the definition of a stem cell to at least some stem cell biologists. But to cancer biologists, some cancer biologists, that's very, very confusing. And how can you call this cell a stem cell? And so that's also, I think, part of the confusion is just the semantics and the definition. And many people are working on different definitions of stem cells. So there's another argument that non-tumorigenic cancer cells can de-differentiate into stem cells. And there's really two models of, there would be really two models of this work by Dr. Weissman and Dr. Jameson. And the first is you've got acquisition of self-renewal properties by some of the oncogenic events in a progenitor cell pool, and it's a cell population that's non-self-renewing, or you have, or these mutations cause the progenitor cell to de-differentiate and become a stem cell. And work by Balaji Akala in our lab, published in Nature last year. What Balaji found is that mutation of 3 genes, which are commonly mutated in cancers, P53, P16-INK4A, and P19-ARF, that these mutations allowed otherwise normal, very early progenitor cells to acquire the ability to self-renew. I won't go into the details, but these cells, as far as we can tell, by all criteria, do not de-differentiate. They maintain themselves as multipotent progenitor cells, but they have the ability, they now have acquired the ability to self-renew. If you look at more differentiated progeny from these cells, they lose the ability to self-renew. So that suggests that the more differentiated a cell gets, the more molecular mechanisms or fail-safe mechanisms are there to prevent abnormal self-renewal. So another argument is that at these mouse assays using immunodeficient mice to transplant human cells are artificial, and that these mice microenvironments only permit the growth of cancer stem cells. And this is recent work from Pierre Delerba, Haipeng Liu, Sam Gambier here at Stanford, who did the imaging using new reporter genes that emit light that Chris Contact developed. And basically what they did is they labeled non-tumorigenic cancer cells, and this is from a breast cancer, and tumorigenic cancer cells, and simply looked at them in the mouse and asked what happened. And what they found was, in the first 2 weeks, the progenitor cells actually proliferate better than the stem cells in the mice. So the microenvironment in the mouse is fine for supporting the proliferation of these cells. These cells maintain themselves, as far as we can tell. We've looked up to 4 or 5 months, so they're not being rejected, they're not going away. They're just simply stopping, they have just simply stopped proliferating, just like a normal progenitor cell would divide a few times and stop proliferating. If you look at the stem cell population, however, these cells, although they don't initially grow as fast, they don't stop growing and they go on and form tumors. So this strongly suggests that at least for this tumor, the mouse microenvironment is fine for the proliferation, for allowing these non-tumorigenic cells to proliferate, but these cells only can do so briefly. This is data from Laurie Prince and Mark Ailes, where they found that CD44 was a good marker for identifying squamous cell carcinoma of the head and neck. And this is immunochemistry, and these green and red cells are the self-renewing cells in this particular tumor as measured in one of these transplant assays. You notice these cells are adjacent to the stroma, where the normal skin stem cells or squamous cell stem cells reside. And these cells generate these cells in the center, which do not, of the tumor, which do not express CD44, do not transfer the tumor. And when you stain with involucrin, a differentiation marker of normal squamous cells, that these non-tumorigenic cells have matured, stop expressing self-renewal genes, and now and look pretty much like normal tissue, although obviously the architecture is not entirely normal. Now, keep this image in mind with this band of stromal cells, the immature cells adjacent to the stroma, and then the more differentiated cells internally. This is still assayed in a xenograft system, so it's still formally possible that there are differences in the mouse microenvironment that don't support proliferation of these mature cells. This is data from York Holskin's group, where they look at chemically induced squamous cell carcinomas in mice. So these are mouse tumors in the mouse. If he stained with CD34, if you notice which is an immature cell marker in the mouse, you notice this, the CD34 positive cells are adjacent to the stroma. These cells are able to transplant the tumor when transplanted in the mice. And then when you look at the cells internally in the nodule, these cells do not express CD34, but instead they express cytokeratin 10, which is a maturation marker in the mouse for differentiation, and then go on to make totally stratified squamous epithelium, and these cells are unable to transplant, to transfer the disease. So again, if you look at the mouse tumor in a moderately differentiated tumor and a human tumor, moderately differentiated squamous cell carcinoma, you see the same histology, the same expression of immature cell genes and mature cell genes, and the same cell populations are able to transfer the disease. So I think this really dramatically proves that you can get tumorigenic and non-tumorigenic cells, and that it's not entirely an artifact of the xenograft system. And finally, work from Rob Cho shows that in mouse breast cancer tumors, in MMTV1 Wnt tumors, Rob was able to show a population of cells which represents about 5% of the cancer cells in these tumors is highly enriched for the ability to form tumors. They form tumors with all the heterogeneous cell populations in these tumors, and this work has been replicated using slightly different markers, but with essentially the same results by a second group in Australia. And then there's been a third mouse breast cancer tumor, P53 mutant tumors, that a group at Baylor has shown in this murine tumors, there is a stem cell population. So again, showing that in syngeneic or identical twin models, the microenvironment, the mouse microenvironment, so where the microenvironment is totally normal and it gives totally normal signals, that this one cell population is highly enriched, and that there's a stem cell population. Now, there's been a recent argument in melanoma that the cancer stem cell frequency is high and is not clinically relevant. Well, if one looks carefully at that paper, what you notice is the comparison of the number of cells that are able to form tumors in these immunodeficient mouse models uses NOD-SCID mice that have an additional mutation that eliminates, which eliminates the ability of NK cells to recognize cells, or eliminates the natural killer cell population. But the comparison in melanoma was done using Matrigel in the NOD-SCID IL-2 common gamma chain mutant mice compared to the NOD-SCID mice. Now, this paper had the lowest number of tumor-forming cells of any paper I've ever seen. And, but there's a couple of points that I'd like to make. First, the comparison is between Matrigel and vehicle. And I personally haven't tested NOD-SCID mice versus NOG mice in melanoma. I don't work on melanoma. But a group at Stanford has tested this. But I can tell you in breast cancer and in colon cancer and in head and neck cancer, if you don't use Matrigel, you do not get tumors. I don't care how many cells you put in, you do not get tumors. So let me reemphasize what I said. In these epithelial tumors, without the addition of Matrigel, it doesn't matter what mouse strain you put the cells in, they will not form tumors, at least in our hands. If you add Matrigel, you go from injecting 1 million cells and not getting tumors to, with the best sorted population of cells in our hands, it takes about 100 tumor cells to form a tumor. Now, the group at Stanford has, then the second point of this is this paper was using cells derived from metastases and cells that had been passaged, for many of the patients, examples, had been passaged in immunodeficient mice. We have noticed when we passage tumors from breast cancer and from colon cancer in immunodeficient mice, the frequency of the stem cells goes up. There's something about the selection pressure, there is a selection pressure that increases stem cell frequency with passage in mice. Irv Weissman's group, I don't have the data, has actually gone back and looked at melanoma, and they have found a stem cell population in multiple patients with melanoma, and using these NOG-SCID, using these same mice, these NOD-SCID IL-2 gamma chain. And again, I want to emphasize that the frequency of these cells in melanoma in NOD-SCID mice or NOG mice is much, much higher than the frequency of the cells than we see in breast tumors or colon cancer. Most breast tumors or most colon cancers. So this is the one of the most aggressive tumors I know of, and probably does have a high stem cell frequency, probably higher relative to most tumors. Now, the data in the mouse, and this is data from our original paper, we found that two things that seem to hold up in most of the tumors that we've examined, including the mouse tumors. And we found, and what Mohammed found was in 8 out of 9 patients, the cells from the patient, the CD44 high, CD24 low cells had most of the tumor forming ability in 8 out of 9 patients. The percentage of cells in these tumors varied from a low of about 2.5% to a high of about 40 or 50%. So huge. So we noticed, and you can see from these FACS plots, these squares, each dot is a cell, and they represent this box represents where most of the tumorigenic activity is. You can see there's wide variation from patient to patient in stem cell, at least frequency of cells that express this marker. And in these 8 patients, these are the cells, as I said, that had most of the tumorigenic activity. He found an exception, though. In patient number, what we called tumor 7, in this patient, the CD24 low, the CD24 medium, and the CD24 high cells all were equally tumorigenic and represented about 66% of the cancer cells. So this suggests in this tumor, the stem cell is different and they're more frequent. And this, by the way, was a patient with gametotype differentiation, and that's known to be an extremely aggressive form of breast cancer. This is work from Piero Delerba in colon cancer. Piero found several markers that work for colon cancer. He found that again, CD44 works. He found several other markers. Again, these boxes represent the tumorigenic population. They represent from 0.6% of the cells to about 45% of the cells in colon cancer patients. Again, huge variation from patient to patient. The average is about 5% of the cells. And in normal colon, he found this very same population of cells that represents about 2% of the cells. And so the percentage of these cells that had all of the tumorigenic activity in colon cancer, the percentage of these cells vary from less than what we see in normal colon to markedly higher frequency than what we see in normal colon. This is immunochemistry of the normal gut, and just if you look here, this is the bottom of the crypt. This is where the stem and early progenitor cells are. They express CD44. So the normal stem and progenitor cells also express CD44. Their more differentiated progeny do not. So the same immature cell population is seen in colon cancer, and it looks like, at least by marker expression, and also by array analysis, I can say, like an immature cell, whereas the non-tumorigenic cells do not express this marker and look more differentiated. Now, another indication that the frequency of stem cells varies from patient to patient and actually is has clinical meaning is work by Rob Cho. He showed that using a gene signature derived from mouse breast cancer stem cells, when he looked at tumors, whole tumors, whose gene expression analysis was measured in patients, in human patients, he found that tumors whose gene signature of all the total cancer cells in the tumor more closely resembled the mouse gene signature, that those patients had a much higher risk of developing metastasis and dying than did patients whose tumors were distant to this. So what this implies is, if you have, the implication is, the simplest interpretation is, if you have a high stem cell frequency, you have a high risk of metastasis. This doesn't formally prove this, but it's very suggestive. Now I'm going to talk about some very basic work that I think has clinical relevance of why being able to isolate stem cells can help in human cancers. So one of the questions, as I said, that we wanted to work on is, what are the molecular regulations of self-renewal by cancer stem cells and normal stem cells? So this is work from Yohei Shimono, which was published recently in Cell a couple months ago now. And what Yohei found was that 37 microRNAs, and these are RNAs that regulate the expression of other genes, that 37 of these genes were consistently differentially expressed in cancer stem cells versus non-tumorigenic cells. And most of these 37 genes were also differentially expressed by normal human stem and or early progenitor cells compared to their more differentiated progeny. So we have this set of regulatory microRNAs. Looks like it's differentially expressed in the normal breast, to emphasize this, and in breast cancer. Now, we decided to work on 8 of the genes which were consistently downregulated in all the tissues. And what we noticed is that these 8 genes were actually only transcribed from 3 primary transcripts. And when we looked at their predicted targets, we found that there was a huge amount of overlap. 5 of these microRNAs were predicted to regulate the expression of very similar target genes. Now, one of the genes that we noticed that they were predicted to target was a gene called BMI1, which is very close to my heart because Angie Park had shown that BMI1 was absolutely required for self-renewal of normal blood stem cells. Subsequently, it's been shown by Sean Morrison to be required for self-renewal of normal brain stem cells, and Martin Loheisen has recently shown that it's involved in maintenance of breast duct. So Yohei transfected normal breast stem cells either with a control virus that expresses this green protein, GFP, and you can see that these normal stem cells form normal ducts, which is what you would expect. But when he infected cells with the lentivirus expressing MIR200C, these cells lost the ability to self-renew. They only form very small foci of colonies, and these cells were differentiated into a myoepithelial phenotype. He then looked at breast cancer stem cells. Now, these are cells isolated directly from a patient, and what Yohei found is these same microRNAs inhibited the ability of breast cancer stem cells to form tumors. So in other words, this microRNA that's regulating the process of self-renewal in normal stem cells is also regulating self-renewal in cancer stem cells. We then went and looked at embryonal carcinoma. We actually submitted a grant to CIRM to look at this, and which was almost but not quite funded. It was we were told we were nuts that this couldn't work, and so anyway, these same microRNAs that are downregulated in human breast cancer stem cells are downregulated in embryonic carcinoma cells. Same genes. They also regulate, these genes have subsequently been shown to regulate by a paper that came out this December, which actually confirmed our results that these microRNAs are regulating self-renewal pathways. And they found, this second group found that these microRNAs are actually also regulating expression of several embryonic stem cell self-renewal genes, and that these microRNAs will appear to inhibit tumorigenic capacity of embryonic stem cells. And then one other example of how this could be useful. And so the point of this is, by studying these stem cell populations and by studying these gene expressions, especially down at the single cell level, that one can look at oncogenic changes. And one of the things that I think Bob will probably remember, when CIRM first started, I thought that there was a high risk of embryonic stem cell lines undergoing oncogenic mutations, and that using these cells, one would, for therapeutic, for regenerative purposes, regenerative medicine purposes, one had to be very careful that they hadn't undergone oncogenic changes. And I think this really highlights that one has to be very, very careful about these cells, and that there now, with these single cell technologies, there's ways to do that. So what about for therapies? And as I told you, at least in some types of testicular cancer, you can actually, platinum will eliminate the stem cells. You have residual cancer cells, but many of those patients are still cured, even though you've left cancer cells behind. But if you have a therapy that leaves residual stem cells, patients would relapse, and that's not the hypothesis. And this is work of Max Dean, who's now on the staff here at Stanford. And Max asked a very simple question: are our breast cancer stem cells more resistant to one of our most commonly used therapeutic modalities, radiation, than our non-tumorigenic cells? And to make a long story short, what he and Rob Cho found was indeed that appears to be the case. When one irradiates a breast tumor, the stem cell population is much less efficiently killed than are the non-tumorigenic cells in this tumor. And this is data actually from a syngeneic mouse model, so there's no microenvironment changes. This is a mouse tumor. He has similar data in human tumors, but I won't go into that. What Max noticed, though, he noticed that the cancer stem cells underwent much less DNA damage than did the non-tumorigenic cells. What Max also realized is that one of the major mediators of cell death from radiation are reactive oxygen species, or ROS, or oxidative stress, and that normal stem cells, normal blood stem cells, had been shown by two groups, first by Toshiya Suda's group and then by Gary Gilliland's group, who elucidated more of the molecular mechanism of this regulation, that normal blood stem cells protected themselves from reactive oxygen species, or ROS2, and that this was necessary for their maintenance. So Max looked at an enriched population of normal breast stem cells and an enriched population of human breast cancer stem cells, and found that the normal breast stem cells and the breast cancer stem cells had, on average, much lower much lower levels of ROS than did either more differentiated cells in either the normal breast or the tumor. He then used a technique, the same single cell technique, which again CIRM thought wouldn't work, and it works. It works really well. And what Max found is that the two enzymes that are rate-limiting enzymes for making glutathione, which is one of the major free radical scavengers in the body, were overexpressed by the stem cells. And the stem cells also overexpressed FOXO1, which is the master regulator that Gary Gilliland had identified. And these are the breast cancer stem cells versus the non-tumorigenic cells from this mouse tumors. And so this gave him a molecular key. And so what he then did is used a drug, BSO, which specifically targets glutathione synthetase, and was able to show that by inhibition of this enzyme that's overexpressed in the stem cell population, he can now sensitize these cells to radiation, and they become as sensitive to radiation as do the non-tumorigenic cells. And so I think this gives you an idea how these tools and how isolation of these stem cells can really give insights into both fundamental properties such as self-renewal. If we hadn't been able to isolate the stem cell population in the breast, in the normal in the breast cancer stem cells, even if it had been 50-50, even if 50% of the cells in a tumor had been tumorigenic cells, we would have not noticed that those cells don't express those 8 microRNAs. And as a matter of fact, the frequency was lower. It was about 5% in most of the tumors. So anyway, my conclusions of this are cancer stem cells use self-renewal pathways that are also used by normal stem cells. I think we've definitively shown that. Some cancer cells use pathways to protect themselves from therapeutics, also used by normal stem cells to protect themselves from genotoxic stress. Max Dean, who's now faculty here at Stanford, has shown that. And I think for many types of tumors, such as leukemia, breast cancer, colon cancer, head and neck cancer, and there's going to be data coming out that even for melanoma, there is a stem cell population. And I think the evidence is very, very strong. Some feel otherwise, obviously. I want to point out a couple of things. Many arguments against cancer stem cells apply to normal stem cells. So every single argument that I've heard about normal stem cells, you could use for blood stem cells. in this room. So you would also have to deny the existence of normal blood stem cells, or skin stem cells, or brain stem cells. So again, these are the same arguments. The frequency, what I won't argue, is that the frequency of cancer stem cells can vary from infrequent to common. And in fact, we showed that and said that in our original paper in breast cancer, and have seen it time and time again. That doesn't, I don't think that says that one shouldn't study these populations and know what's going on in individual patients. And the last point is, or one of the last points, is the study of cancer stem cells still has therapeutic implications even if all the cells can drive tumor growth by one of these other models. So if the cancer cells really can dedifferentiate, and in science one can never prove a negative, I mean, that's an experiment. You can only prove dedifferentiation if you see it happen. We've never seen it happen, and we've looked hard, but that it might happen. I'll be honest, you know, I can't totally ever exclude that possibility. But these cells, if they dedifferentiate, they have to go through a stem cell state where they're using these maintenance pathways, these self-renewal pathways. So a therapeutic, if you develop a therapeutic against a self-renewing cell, even if all the cells can eventually dedifferentiate into this state, that therapeutic will still work. So again, for practical reasons, it doesn't negate the power of studying the stem cell population. And it's also clear that this cell population is often not responsive to some of the therapies we use. And so developing therapies against this subpopulation of cells, which is clearly there in tumors, could be combined with therapeutics against the other cell populations. And I think the other thing, and the thing that excites me most, is that cancer stem cells actually do probably arise from progenitor cells in many cases, maybe most cases. And this suggests that because these progenitor cells respond to different growth factors, different mitogens, and different adhesion molecules than do normal stem cells, this suggests that by understanding those differences, one could really specifically target this self-renewing cancer cell population while sparing normal stem cells, because an agent that kills normal stem cell too is going to be too toxic. Nobody will live through it. And with that, I'd like to acknowledge the folks who did the work, and I think I mentioned them throughout the talk. So.
Okay, thank you very much. I think at this point we need to thank Dr. Clarke, and we will commence our normal meeting immediately. Thank you very much.